All right, so let me start. So welcome to the first event of this semester's CAU Cinema and Media Studies Curriculum, a series of invited talks aimed to promote and institutionalize cinema and media studies in South Korean academia and probably elsewhere. So today, I'm happy to host a talk by Dr. Bishnupriya Ghosh. So let me introduce her. Dr. Ghosh is a professor in the Department of English and Global Studies at the University of California, Santa Barbara, also affiliated with comparative literature, film and media studies, and feminist studies. She has published many articles in prestigious journals, including Jump Cut and the Journal of Post-Colonial Studies, Postmodern Culture, and a number of important edited collections, including Sustainable Media, co-edited by Janet Walker and Nicole Starosiewski. And she's the author of When Born Across, the literary cosmopolitics of the contemporary Indian novel, published by Rutgers University Press in 2004, Global Icons, Apertures to the Popular, published by Duke University Press in 2011. She's also a co-editor of the Routledge Companion to Media and Risk, co-edited by Bhaskar Sarkar, published by Routledge in 2020. And recently, she has published the tiny and fascinating book entitled The Virus Touch, Theorizing Epidemic Media as part of Duke University Press Experimental Future Series in 2023. Today, so this is the book, and today she'll give a talk related to this book entitled Multispecies Politics in the Epidemic Episteme. So please welcome Dr. Bishnupriya Ghosh.
Thank you, Dr. Kim. This is a real pleasure to be at your colloquium and the first of them. Let me share my screen first before we start. Yeah, if you can see that? Yeah, okay, all right. So, as the introduction tells you, one of the questions I always get asked is, how did a post-colonial studies media scholar get to write this book? And it started in a kind of an odd space in the sense that after 9/11, a group of us were very concerned over how the new surveillance technologies were targeting certain groups. And so we began to think about the relationship between media and risk. And that's where it started. And one of the things was my generation had a huge number of losses in the HIV/AIDS epidemic. So my portion in that project was to think about biological risk, right? Communities that were targeted as biologically risky. And with my base in South Asia, I had been, working with HIV/AIDS organizations there. What was very curious to me is I was hearing a very different story in the global South than the one in the US, because in the US, the antiretrovirals had come. It seemed like, the pandemic, it had become a chronic disease, so to speak. But where I was working, there were migrations, people were not getting access to meds, so that it was really what living with HIV meant was quite different. So I really started there, and I began to think, how do we perceive epidemics? Because this is really about media centrally. You can't see viruses. You don't know how many people are infected. So how do we actually perceive epidemics? And that's where this whole project around epidemic media really started. And interestingly, as, oh dear, my PowerPoint doesn't seem to move. Okay, yeah. Interestingly, as I started on this project, it was a very difficult book to finish because a second pandemic broke out, right, in 2020. And I thought, well, am I the only person who's writing on HIV/AIDS when COVID is all around us? Turns out there were 75 books published on HIV/AIDS during the last 3 years. So, there's something to do with the HIV/AIDS era as a kind of an archive or a historical lesson. So I want to today really talk about epidemic media. And this is one of the first times that we actually see a virus, the tobacco mosaic virus. And what I was very interested in is how this is a kind of a media form. So micrograph is a media form that technically and aesthetically composes multi-species relations to render it intelligible, right? So you're thinking about the viral particle in a certain medium, host medium, and that itself, it's a relationship with the gene as a kind of privileged form of life. Now, what happens, one of the sites that I visited, one of the things that I thought I'd explain this by is the PCR test, because almost everybody had a PCR test in the last few years, right? And what the PCR test tells you is X amount of virus in Y amount of blood, right? So there is this object and environment constantly composed, and that's a numerical composition. So one of my research sites was this clinical medicine lab, which actually prepares a blood specimen, becomes several aliquots or samples. And then on the right, you see the PCR machine, right? Which is not really your traditional media technology. But I was thinking about how are these technology actually telling us, taking blood and converting it into a number. So I began to look closely at that. And what increasingly became really interesting to me was, because you're looking for a particular virus, identifying, classifying, and counting it, there are many, many other cues in the same blood that you don't see. And to this seemed to me, following someone like Jacques Rancière, things that are partitioned away from the proper composition, X amount of virus and Y amount of blood, those began to interest me because those seem to me to tell you much more about multi-species relations than the PCR test itself. And I'll explain this as we go on. So one of the things that was very clear to me, that despite the fact that you were looking only at a number of viral particles, one of the things is a very bad picture of me in the lab, but one of the things that was, it's not just this formal composition, right? It's not just a vial with blood in it. That it's also embodied practice and all the safety protocols, the hazmat suit, the gloves, and notice it's also tuned into HIV/AIDS, right? I'm not wearing a mask. This is 2017, right? There's a sense of haptic danger. So I began to think, well, blood, saliva, feces, all of these vital samples are really, you think of the body as the site of origination. But one of the things that we do is we are afraid of them. We are apprehensive, right? There's anxiety connected to them because they breach the epidermal limit. They move between bodies. And if you think of all the blood donation controversies of the HIV/AIDS era, that was really about blood going from body to body, right? Crossing species and crossing between hosts in a species. So this sort of thinking made me think about these vital mediums and elemental mediums like air, water, soil more expansively as really what tells us about all the multi-species relations that we live with. And of course, by now, I don't even have to tell you this. When we talk together in person, we see the air between us, right? We've begun to see medium, right? So this is where I started. And one of the things that I will talk about in a minute, but let me just finish up on the major sort of architecture of the book. You'll see chapter 2, 3, and 4 are the media chapters. One is about the image, right? How the image of the virus is made, visualization. The second is about the medium, right? What is the medium that's tested? So it could be blood, it can be saliva. Today, I'll talk about feces. And the last is where I'll go today is about the animal host, right? So one of the things that we know is that animals actually transport viruses because viruses famously have no locomotion. They can't go anywhere on their own. They hitch a ride on mediums, right? So the 2, 3, and 4 are the main media chapters. And then I have, importantly, a chapter 1, which is a much more historical chapter. And this historical chapter was important because this was a history of how we began to think about pandemics, right? And this is sort of in the early 1980s, there's a sudden outbreak of Marburg, HIV, Hanta, Ebola. And people begin to think that, have we lost the war on germs, right? And that's sort of what I call our current epidemic episteme. And I'll return to this point later. But let me finish off by talking about epidemic media, of the two things you saw, the micrograph and the blood sample. I am interested in a mode of perception that we called apprehension. Sensory, affective, intuitive apprehension complements and sometimes unsettles our rational recognition or comprehension, right? So we arrange a proper study, we recognize some things, and then apprehension, we sense, right? And one of the things that triggered this is when I was at the clinical lab, one of the technicians said, well, you know, we only deal with molecules like viral particles. That's the scale of analysis. We're not really bothered with multi-species stuff. That's all about organismic scale, right? It's about species. But she mentioned something interesting that I found in my interview transcript. She said, you know, we read a sample of blood this morning that I kept thinking there's some error. It can't have this much virus. And she said to me, it had so much virus in it, the sample could have walked on its own. And I thought, what an odd thing to say. It is almost as if the blood sample, had become another animal. It had its own agency and it was going to walk on its own. So in this way, things that were irrelevant to the study kept bringing back this deep conception that we are always already multi-species, right? So that was the beginning, and I began to think of, well, you can be apprehensive of all of these, you know, pathogens around us, but you can also kind of think about apprehension as a creative process that seizes upon the whole based on partial knowledge. That is, it is an intuition. That's what intuition is. You know something little, but you try to imagine the whole from it. And so I was thinking how all the noisy cues that we don't look at, that we try to partition away, actually lead us to this larger world of multi-species relations that I will call the living milieu. So let me go a little bit into multi-species studies. One of the things, this is the cover of the book, of course, right? This was an image made by Luke Jerram called Past, Present, Future. It's got variola, that is smallpox, HIV. And in 2015, he made a future virus that kind of looked like SARS-CoV-2, right? And this was kind of interesting to me because, of course, we are always trying to imagine what's coming next, the pandemic to come, right? And he had already imagined this in 2015. So I was thinking about how COVID put the zoonotic transmission between humans and animals on the table in no uncertain terms. Many of the anthropogenic drivers of pathogenicity, so things like deforestation, industrial farming, illegal wildlife trading, all these things had been clearly identified by the early 21st century. But this intuition that there is a bigger picture, that we really should be looking at a multilinear sort of emergence, is very old. 1855, Rudolf Virchow put together Zoon, that is the animal, and Gnosis, disease, and he basically called it zoonosis, right? Diseases that skip from animals to human. And he argued at that point, 1855, that human health couldn't be disconnected from the human-animal interface. Despite the fact, at the time when he's talking about this, people were talking about germ theory, which is one pathogen and one disease, right? He's saying, no, we've got to have a much more multi-scalar understanding of disease. If we only focus on human symptoms and human pathogens, then pandemics will keep coming. We're not attending to the right thing. So this was his one medicine formula. And it takes more than a century for that to come into being. So by 2018, the World Health Organization had adopted what was called Structural One Health, and disease X with pandemic potential had become a top research priority. And that's why I showed this picture, because of course that last virus is disease X, as Luke Jerram, this artist, had imagined it. So this scenario it really comes about in the early 20th century because of the sudden emergence of deadly viruses. Now, if you remember, in the post-World War II, the mass manufacture of penicillin, right, seemed to be, at that point, it had been manufactured for the US troops, right, who were fighting a war. And there was this sense that, wow, we're really going to win the war on germs. Excuse me. And that perception, it actually climbed up. By 1978. We had eradicated smallpox. And it seemed like we had won the war on germs. And then right after 1978, early 80s, you have HIV, Ebola, Marburg, and Hanta come up, right? So there's this utter melancholia. Excuse me, I have a scratchy throat today. But by the early 80s, you have this sense that you have these microbes that are going to come out of the recesses of the planet. They're going to skip the species, and then they're going to enter human populations, right? And Laurie Garrett, of course, wrote a really famous book called The Coming Plagues that sort of caused a kind of widespread panic. This is the point at which we begin to see multi-species studies come about, right? And this is, I want to distinguish multi-species studies from animal studies, because multi-species studies draws not attention to large charismatic animals, like tigers or dogs, or, these animals that, say, Donna Haraway has written about, but they draw attention to kind of unlikable pests and pathogens. And they also, important part of multi-species studies, they don't separate it from water, soil, or air. So the biologic and the geologic are often thought together. They talk about how, our gut bacteria, etc., we are all rhizomatic formations, right? Except we keep cutting and saying, I'm a different species, I'm a different individual, right? So they question the boundaries between species and say these are crafted, these are epistemic cuts. And what do we do when these cuts arise? What's the consequence? This is why I'm interested in epidemic media in the way I've described it so far, because one of the things that you do when you cut out a blood sample and say, this is your blood and your PCR test, it's very important because it's your state of infection. But this sort of medical cutting often obscures the long view, which is thinking about habitat degradation, animal health as human health together, right? So multi-species studies actually really gets there. The second thing about multi-species studies that I'll say is that there's a bunch of biologists, sociologists, and geographers who criticized the over-enthusiastic embrace of multi-species flourishing. They argued that the abundance of awkward creatures, such as pests and parasites and pathogens, make multi-species togetherness difficult when vulnerability is in the making and death is at hand, right? So they say that the state of infection is really when one kind of species flourishes at the cost of another. And usually we tend to see this as a state of war. And I will show you this video without playing sound. This is a classic way of approaching pathogens, that they are the invisible enemy and we are at war with them. This was a public service message, HIV message that, so you can put in any country For instance, you can put in South Korea instead of India or China, or, it was sent to Botswana. So, and you'll see how territorial this understanding it. It's pathogen and me, right? Nothing else is involved in this diagram. Okay. So multi-species studies, I'll let this play while I talk. Multi-species studies then begins to think about this rhizomatic formation where we are already multi-species. There is nothing like this body that is there's no inside and no outside in some ways, right? And they say that quite apart from this idea of just being with all creaturely life, let's think about entangled materiality as a problem. How we navigate this can be a return to unthought anthropocentrism, but that's the very state that got us in this sorry space, this idea like just kill and exterminate all germs. Well, we haven't won that war. You think of the history of pesticides. You think about anti-resistance, the bacteria that are resistant to medicines. The killing and extermination of microbes has not worked as much. So how do we approach our multi-species being? This is the question. So what would this politics then, after the late 20th century, look like for our present pandemic circumstance? These days, we have bats and raccoon dogs on our minds, much like our obsession with wild primates in the last decades, or with rodents as plague agents in centuries past. Epidemic media track animal reservoirs. Okay, this is different kinds of epidemic media that track animal hosts, who are intermediate hosts, they're at reservoirs and they're sentinels, and we're hyper aware of how they migrate, how they get sick, their health, their populations. So reputable emerging infectious disease models suggest that 60.3% of these emergences are zoonotic, and among them 71.3% originate from wildlife. The stories of anthropogenic ruin illuminate a colonial sphere of ongoing catastrophes, alerting us to new contact zones. Bats forage beyond their home range, rodents come in contact with farmers, the bacteria and viruses that they bear enter new hosts. It is clear that the best investment in epidemics is to develop a transdisciplinary multi-sectoral approach that cross-hatches human health, animal health, and ecosystem health. Such an orientation does not actually privilege, and this has been the critique, it doesn't privilege animal health over human health, for we know that environmental degradation is unevenly distributed across the world, right? Some of the most vulnerable communities face it. And colonial practices of conservation often put the well-being of animals before that of humans. What is to be done then? And so let me turn now to this particular biogeographic area of Bangladesh. It's an area in South and Southeast Asia that didn't make it into the book for all kinds of reasons. But as you'll see, the work that I did on the animal hosts of HIV/AIDS was absolutely key to this particular study. One of my guides was a wildlife biologist, Anne Laudiceau, who had trekked for many years in the Ituri Highlands bordering the Democratic Republic of Congo and Uganda. And it was particularly relevant because, of course, this was the region where HIV-1, the M group that has caused the global pandemic, had emerged. And she was following actually a bunch of relict monkeys who didn't have much contact with others. So she was very interested in seeing how they carried SIV. So, there's a strain, of course, of simian immunodeficiency virus, right? And the monkeys carry it just fine. It's only in the crossover to the humans that it becomes pathogenic. If you see the straight lines there in that diagram, those are called transects. You follow that line and you sample everything in the habitat on two sides, right? So nests, water, soil, insects, I'll talk, of course, about fecal matter. And so I was very interested in this idea of how do you sample a habitat? right? You can look at the viral gene, you can look at the human body, but how do you actually make sense of a habitat? So I really followed her in thinking about how to think about this multi-species question. And I had come across her because of my contact with EcoHealth Alliance. You may have heard about this. You know, the Peter Daszak, the guy who was working with the Wuhan Institute of Virology, is the CEO of EcoHealth Alliance. And I was not really interested in him, the celebrity scientist. I was interested in the kind of work they do all over the world in predicting viruses to come. And my main interest in them was they try to create these alliances between global research institutions like the Wuhan Institute, universities, local laboratories, farmers, wildlife experts. And so they try to build capacity within communities, right? So this is how they map. You see the first two are rodents and bats. They're some of the highest hosts of the many, many viruses, right? And so they chart it according to how high the risk is. Now, the particular piece I'll focus on today is really, as I said, in Bangladesh. This was a study between the University of Washington's National Primate Center and the Jahangir Nagar University of Dhaka. And it takes, you see these marked out places, it takes 2 forested and 4 urban regions in Bangladesh as the sample, right, for viral strains carried by the non-human primates. And so there's a great deal of interaction because of the habitats, or because of deforestation. There's more and more interaction between humans and monkeys. And so I was interested in this EcoHealth Alliance study about these monkeys in this region. Now, one of the things that comes up here is that the monkeys were entering into human-altered environments, and so they were interested in tracking how the virus in them would mutate. So actually, let me say a little bit about what they ended up doing, and it'll give you a sense of what I mean about this kind of multilinear approach, right? They ended up doing something rather simple. In one of these places, they found that the government had instituted a program where, you see the wall here, it's sort of the edge of the forest. They sort of planted a buffer zone of fruit trees. And what that did was the monkeys actually were just coming into human backyards, and they were coming into agricultural land because they wanted food and water, right? They'd lost those sources. As soon as they planted this buffer zone, the monkeys didn't come in contact with humans, right? So it's a very simple, pragmatic solution. It was built with the local communities, right? But this is sort of the point, that how is it that we can imagine a way of putting some distance between certain kinds of probably contact that we'd feel apprehensive of, right? How can we anticipate this? And to anticipate this, we actually have to think about this larger living milieu that we all share. So again, the HIV story is actually really, really interesting here, okay? And this is a complicated story, and let me read it to you. We know that the simian immunodeficiency virus was tolerated by wild primates that existed in Cameroon for 100 years. But it became pathogenic in its zoonotic spillover to humans. Epidemic media on HIV strains are extensive. So if you see on the left, that's really, a micrograph, right? You take a sample from someone and you look at HIV particles under the microscope. The one on the right is what is called a phylogenetic tree. It's a data visualization. And you can see that on top left is HIV-1, right? And you can see the many strains, A, B, C, D, F, G, okay, that develops. And that's the one that created the global pandemic. So they actually look at how viral strains mutate. So one of the things that they found is really that the horizontal gene transfer that transfers from co-infection between monkeys within a given area enables the viral evolution, displacing the logic of biological descent. The jury is still out on tracking the original reservoir for this SIV strain, right? The pathogenic one. But what the contamination story does, the co-infection story does, is to embed the genetic mapping within ecological relations. The environment is no longer just this particular monkey's vital medium that you see on the left, the blood or saliva or fecal matter, but also the simian population's habitat. In those habitats, phylogenetically proximate mammals mingle, and this is important as a factor for spillover. So think about it from the virus's perspective. You're going to look for environments that are similar to the one that you live in, that you find sustainable. So if mammals that are genetically similar are easy, you're going to skip from one host to another, right? So there is this crossover between monkeys and humans, because humans and monkeys share 93.5% of our genetic makeup are the same, right? So you can see why primates in particular are one of the carriers, right, of new viruses that we may not have encountered before. And we don't know what that those will do in our body. So typically, tropical forests are densely populated with diverse species, becoming more so. So they're more and more dense because of deforestation and habitat degradation. Hitherto, non-human primate populations concentrate in fewer and fewer sustainable habitats, and the threat of co-infection between them increases. Recombinant strains emerge as viruses mutate to stay alive in both species. So this, and then of course you have this destruction of the habitat, nesting niches, water sources, and food. The wild NHPs and humans live in new proximities, as you saw from those photographs. This is the complex HIV story, and it's a roadmap for other stories of receding fruit and water sources in all 6 areas that generated more physical contact in this Bangladeshi instance. So what I'm getting at here is, if you're going to think multi-species, there's a kind of a non-linear temporality of the spillover. There's a time of virus. When is it going to mutate? There's a time of the primate. When is it going to move? When is it going to age? And there's the time of the habitat, right? How is it degrading? This means that any origin story that we tell is going to get it wrong. And you know this from the HIV archives, right? The tale of, oh, it came from a green monkey. It came from a patient 0. There's a famous story in this part of the world where there's this hunter that came from the depths of the Congo, and he sailed up with a cut on his arm. And that's what started HIV AIDS from Kinshasa. So these origin stories are counterproductive, because you actually can't track a single causality, right, to emergence. Rather, we are forced to think in these non-linear ways. Posing the multi-species question muddies these causalities. Viruses move between hosts in habitats disappear, new multi-species assemblies appear, all at varying tempos. Multi-species thinking then approaches pandemic emergences as upheavals in multi-species relations, unfolding in this larger living milieu. And this living milieu, of course, I take from Georges Canguilhem. He's, of course, Foucault's mentor, right? And Georges Canguilhem is a doctor writing in the mid-20th century, and he sees the living milieu as no abstracts. You know, when we talk about the planet or one world, these are abstract spaces. He says that the living milieu is a medium. It's full and it bristles with chemicals, with other microbes, with soil, with water. So the milieu is a kind of a medium. And we have a constant debate, right? He calls us living being. He's a doctor, right? So he's concerned with the patient. Living being has a constant debate with their milieu, and has to constantly make new norms as things change. Now, if you think about the infection, what has happened is there's a new kind of multi-species relation, right? And that's what we have to adjust to. Yeah, it's a living milieu we have to adjust to. And we make new norms. How many new normals did we see during COVID? Again and again, we made new norms. So that's why I think Cauccms' idea of the living milieu as this bristling medium that we live in, we see the air around us all the time now. For him, it's a perceptual space. It's a space that we perceive. That's why epidemic media is so important. Media technologies enable us to see the space. And there was a lot of work done that you guys may know in visualizing sneezes. So how far does a sneeze travel, right? To look at that is something that people did. So let me close with a little bit on this particular area. So the region that I was looking at, right, they map viral strains according to geographic regions. So they're already alert to the living milieu. 3 retroviruses are present among simian populations here. One that is most studied is called the simian foamy-virus, SFV. And what they're really, really interested in is how it's not yet pathogenic, right? But they've been watching this Bormi strain. If you look on the left, the Bormi strain has the most variation here, right? And it's quite interesting because there's only one kind of epidemic media, right? So they're following this strain and its mutations and trying to predict that what's going to happen to this strain as it enters other habitats. And this is what has happened, right? So you see the Bormi strain in red there, and Bormi is right there in the middle, but it has traveled, as we shall see, up into the Dhokola region, right? It has traveled into other regions as well. So one of the maps that looks like this, so these are the 3 media, right? You have a phylogenetic tree as one kind of epidemic media. Then you have this kind of cartographic understanding. Where are the strains? And then you have, this is a classic heat map, right? And you see Bormi on the left, the green stuff, right? The red marked stuff. You'll see that the Bormi strain has entered into every other place, especially the Dhokola, right? So the Bormi strain is traveling. Why is this important at all? Well, it tells you that the animal host is moving, right? So that the monkeys that bear the Bormi strain, they're moving into other habitats. So you begin to ask questions, right? So you begin to ask, why are they moving? What's happened to their habitat? What's going to happen if they co-infect each other? When will they come in contact with humans, right? So this is the kind of media study that we can get from, if you follow the cues that these kinds of epidemic media offer. So how were these visualizations made? Really from one kind of sample, not blood like I started with, but fecal matter. And this is, of course, a part of Anne Laudisseau's video from the Democratic Republic of Congo, where she says some of what they collect in the forest are fecal matter that the animals leave behind. And this is really important. Even during COVID, fecal matter became a thing, right? They were, in the US at least, they were looking at fecal matter from dorms, for instance, to look at the level of COVID infection. So fecal matter is preserved in pellet form, then it's analyzed for viral strains. But what's interesting about fecal matter is that's not all you get. You get things like seeds, insects, so on and so forth, so that you might actually get multiple things, right, from a particular kind of fecal matter. A molecular geneticist will pick the viral strain. Yeah, but a habitat, an environmentalist will probably try to think of the animal's diet, right? Will try to think about the animal's age and so on and so forth. In other words, any sample like this is an open-ended media form that has multiple signals. And usually we try to pick out one, a value-laden signal that tells you within a composition you produce. But then if you follow the noisy cues, if you apprehend other things that are in the sample, you can actually build out, or you can intuit the larger living milieu, right? So mind for multiple signals, bristling with noisy cues, the vital sample is an open-ended, taking the curious beyond the gene as a privileged form of life. Apprehension cues you into the host's health, to soils and plants, to insects and other microbes in the entangled agencies of the living milieu. To intuit this whole from a partial traces, and I'm back to the micrograph, is to give over to more questions. Is the forest still populated with the animals who left the fecal matter behind? Are their diets changing? Will they move in search of resources, shelter, food, water? As transport for viruses, what will they carry with them? To ask these questions is to push beyond the proper composition and to speculate on the perceptual space of multi-species relations. In all these ways, epidemic media can be crucial arbiters of our difficult and inescapable multi-species condition, teaching us to unlearn anthropomorphism and to learn being otherwise. The historical awareness of zoonotic changes orients us towards the causal relatedness of all things. As pandemic survivors, we feel life forms unfurling around us, sympoetically, as Donna Haraway once put it, uncontainable, stretching beyond our epidermal limits. Most of this time, this apprehension is scarily diffuse sense of infection. We've all felt this. Infection is everywhere and all the time. But it can also impel relational understandings of life so foundational to the environmental imagination. So thank you.
Thank you. Thank you for your wonderful, rich and brilliant talk, Bishnu. So, before posing my questions related to the book and your talk, so let me give you my impression. The impression on what on the contribution that the book makes to the media studies, the broadly, particularly in humanities, the perspective. So one of the things, one of the most the important things that I find from these books, methodology and object, is not simply about the urgency of its topic. So amid the pandemic situation and the HIV, the spread as its precursor, but a kind of the very the composite kind of methodologies during on the media studies and science and technology studies and the multi-species studies and the medical humanities, etc. In particular, I was so impressed by the ways that you incorporate, the line of new materialist thought illustrated by, Dana Haraway, Karen Barad, the Bruno Latour, and others. Particularly, Karen Barad's influence is evident in demonstrated by the use of the concept of epistemic code and entanglement, as also your very meticulous, investigation into the different labs and different medical, tools and instruments across different regions, including remote sensing technology for tracking animal hosts and the movement of animals in Africa and Southeast Asia. So definitely recalls, Latour's, the investigation of the roles that laboratory play in constructing scientific facts and knowledge, in terms of the ways that laboratories are built. Laboratories are premised on the multidimensional network of different, human and non-human actants. The agents are including objects, including blood samples, and the biological and pathological tools, and a number of different specialists concerned with visualizing objects and turning them into knowledge and fact. So definitely, it's kind of the how the new materialists think. The concept, including actor network theory and compositionism proposed by Bruno Latour, as well as Kahlenberg, entanglement and agential cut, are productively applied to expanding or reconfiguring what media is, what media are, and how they exist and how they operate. So I think this is a very important, and very insightful contribution to media studies in the context of the North American academia that strives to rethink the ontology and operational media by drawing the concepts of element, atmosphere, milieu, and extraction, etc. So that kind of the theoretical project that reconfigure the definition of media with the concept of epidemic media definitely is very informative and very suggestive to particularly the Korean humanities context, because to briefly explain what media studies is regarded in our context. So the media studies is in Korean academia is the primary considered as a kind of social science and communication studies thing, not in the humanities actually. So even though some humanities scholars, such as English studies, English or comparative literature scholar approach the object of pandemic or the instances of pandemic, virus, and etc. For example, they may start with taking such theorems as outbreak or contagion and apply some kind of new materialist thinking to how the theorems represent by the spread of virus and visuality of pandemic and its influence on human bodies, etc. But this is not the kind of definitely that kind of approach is maybe helpful. It's some helpful to explain the public imaginaries of what the virus is and how it impacts our bodies and psyche. But this is definitely, we know that this is definitely so subject to many limitations in the situation where many unknowable, invisible, or unintelligible objects and courses in our environment compel us to rethink what media is, and compel us propel us think of media in relation to a variety of different mediations in different scales. So definitely the book's most important contribution to is not only not only its amalgamation and incorporation of different methodologies, but also the ways that the concept of epidemic media make us, multiple initiate multiple dialogues between media and mediation. So that's definitely so that's the reason I appreciate the book with great care, much care and interact. And the first question I want to pose regarding the book is how is the politics of multi-species relations that you propose throughout the book, including the book's chapter 4, related to the kind of the biopolitics that was primarily understood in the context of Michel Foucault and George Agamben. And to put it differently, my question is, in what ways does the multi-species politics, so prepare us to rethink or expand the concept of the biopolitics in our situation? So, so in thinking of these questions, so what strikes me is Benjamin Bretton's book, The Revenge of the Real, published amid the pandemic. And he also discusses the variety of instruments and technologies, including there were operated to control the virus and the govern people's life and health, and in doing so, he criticized the normative the assumption of biopolitics as a kind of technology for controlling people's the populations and create a kind of the people's subjectivity in more severing ways. He calls it the negative biopolitics, but he proposes in the context of the ways in which the various technologies are deployed to visualize and make sensible the virus and its impact. The use of this technology is not simply about surveillance, but also about making our lives sustainable in relation to other human beings and other species. And so he proposes it as a kind of the positive biopolitics against the negative biopolitics that assumes the use of controlling and monitoring technology as a kind of extension of surveillance technology. So so I think so the Brettons take up the epidemic, the roles of technologies on refiguring the society from the perspective of what he calls the epidemic perspective is closely related to your work on theorizing epidemic media and your emphasis on the multi-species politics. So as a kind of new, so I think that kind of conceptual take allows us to rethink the operation of biopolitics in our situation, in the ways that biopolitics is not simply about the human beings and societies, but also rethinking societies and human subjectivity itself in relation to other species and other life forms and other environmental elements. So I was wondering about, so your thought on the relationship between multi-species, politics and biopolitics.
Thank you for that amazing characterization of the book. That's really, it's pretty comprehensive. And let me take up a couple of things. Also, because I didn't touch on a lot of it, right? I went to the last chapter and the multi-species question. There are a couple of things I'll say here. One is that there's a whole lot of writing by Heather Paxson and other people who talk about cheese making. Yeah. And they actually talk about, I was getting to it a little bit in this talk, but not enough. They talk about the fact that we always govern microbes. If you're making cheese, right, this is a very old art, right? And there are many, many old arts of managing microbes, right? It's not about just living with them or making them kin, but no, we govern them. So she calls it a microbiopolitics, right? And you're right, it goes back to Foucault's, I think his lectures in Security, Territory, Population, where he talks about three events. He talks about wars, famines, and epidemics. Yeah, as these sudden catastrophic events in which the vital life of human beings and human populations are under attack. And then, so he's talking, of course, about the vaccination campaigns and so on and so forth. But Foucault, he doesn't really talk about technology, right? He has the word technology, but it's not media technology in the way that we understand it, right? And so I think Foucault for me is always a starting point, because he says, this is a different form of power, this security paradigm. Yeah, it's not about executive power, cut off the head, right? It's not disciplinary power, send them to the jails and the prisons and the clinics, but it is security that lets things be, but regulates it. And that, I think, is very productive in the sense that if you just stop everything, all kinds of movement, right, human populations will not survive. So you kind of have to let people exchange goods to live and to love and to have sex and have families. You have to let vital circulations be, but you also have to control them. And epidemics are extreme instances where you're trying to control them, say, with vaccines, right? So I think he has that paradigm of security, and I think of that as a very early way of thinking about microbial biopolitics, right? I think what has happened in media studies is that after, John Durham Peters, after a lot of, people who've been writing about ecological media, we now have a much more expanded sense of the medium. I was trying to get to it with milieu, right? That you have air and water, and these are media, but you also need technologies to see them, right? So it's media ecological and media technological at once. So if you go from Foucault to that, then the media technologies, and then the Benjamin's book is actually really interesting that way. But then what role do technologies play in this act of securing life, right? And what kinds of possibilities do they offer as well? It's not just securing, right? So in the chapter that I was talking about, I used the idea of camera traps. You put these camera traps in the forest, and in the camera traps, you catch this movement of animals. It's literally capture and secure to understand. But again, there's a lot of stuff in the living milieu that gets captured on camera that you didn't expect, right? And that's the kind of media reading I'm interested in. I'm following my colleague Greg Siegel, who, his example is the black box, that sinks to the bottom of the ocean when a plane crashes. And you're looking at the black box for certain signals. What exactly happened? Was it an engine failure? Did something hit the plane? What exactly happened? Those are the valuable signals, but you also hear people, screaming. You hear people calling their beloved the last phone calls, you hear a lot of other things, noises that seem irrelevant. But for Greg, that is the condition of existence for the signal. So this media paradigm that has come out of thinking more broadly about media is sort of signal and noise, right? So the signal and noise paradigm in media studies, I think, is really productive to multi-species thinking, because we are not always thinking about it. We try to arrest it. We try to capture it. We try to limit it always. Like, think about this whole thing about, you know exactly who gave you COVID, right? That's a myth in some ways. You kind of think, right? You're always trying to track it back. So this idea, or that, the virus came from the Wuhan lab. I mean, that's a very comforting thought. Humans created it. Much more difficult to think, oh my God, we went mining somewhere or there's deforestation, because those kinds of activities are far more harder to control. So I think multi-species thinking is a complex way of thinking about our relationship to the environment. And so I'll stop there. I can say a little bit more about, the medical part of this, because I see the book, as you said, Professor Kim, in a good way, that it's between science and technology studies, environmental media, and critical global health. Those are the three sort of fields. And that's why the book is so multidimensional, because epidemics are biological and ecological, and they're social and political.
Thank you. So, all right. So, I'd like to open the forum for your comments and questions to Bishnu. So, please, turn on your video and provide your questions or comments if you want, or if you may also put your questions, comment on the chat box, so that I may translate, if it is in Korean, translate it to Bishnu for her reply. So, yeah.
So while other people are thinking of their questions, I have two questions that are much simpler and basic compared to the questions posed by Ji-hoon. But I really wanted to echo what was said before about how this really opens doors, brings down the boundaries between different disciplines. And for me, it really exploded my mind about the different kind of ways that we can think about media, but also the different kind of ways that we can study media as well. On that note, you also wrote in your conclusion of how this might be meaningful, or how to bring it back to media studies, because it is this really vast resource of information and data and disciplines that you're traversing through and bringing together in your book. I was a little curious about your methodology of field work and ethnography and your visits to the labs. And when we think about these labs, we usually associate them with biology or chemistry and computer engineering and things like that. But I was wondering how they responded to your work, and how the kinds of collaborations that really sprouted from this interdisciplinary research that you were doing in a very pioneering way. So that was my first question. Again, just to piggyback really quickly, so the possibility of collaborative research that we really talk about a lot, but a lot of people aren't doing in a very practical sense. So I was just wondering about your thoughts on that and the possibilities that you see. The second question was about kind of going back to what you were saying earlier about your not only your intellectual curiosity, but also your drive, right? Your personal drive to think about how we are the components, right, in this multi-species matrix. But I was just curious, and perhaps this is a personal question, but I was just curious about writing this book, and of course, I'm assuming that you're continuing this research, but I'm just wondering if there's anything that shifted in your own perception of your position or your role or your interactions within this multi-species matrix.
Always the great questions. Hey, Jean, thank you. Let me start with the first one, which is actually opens up some things that I haven't said in this talk. And the first one has to do with the method, right? That how did I actually choose it? And it's interesting, actually, you know, I was telling you the story of how I was working with these HIV/AIDS organizations, right, in South Asia, well before the book happened, right? And in each case, in each of the chapters, I would say I was reading a lot of the literature. I had got the frame in my head that you've got to look at the molecular scale, the clinical scale, and the ecological scale, right? And then I began to sort of think about, well, what is the HIV story here? And one of the things that I started with were these small clinics in the global south, in which these health counselors would try to explain your blood picture to you, right? So there's a work of translation that's happening. And then I began to talk to them and they were like, oh, you know, this local lab actually does the blood processing. So then I went to the local lab and they were like, well, you know, it's MSF or the WHO that funds this. But, you know, we actually get our instructions from, these major labs in the US, right? And so I actually my method was to follow something on the ground practice. I realized there was a difference between that and expert knowledge. And I tried to thread it back, and then I got to this lab in Seattle, which had been dealing with HIV/AIDS blood since 1985. And I have to say, Hejin, the scientists were amazing with their patience, the way in which, they've at first, they were like, oh, are you thinking about HIV/AIDS messaging? And I'm like, no, not really. I'm thinking of the PCR machine as a media technology, because it takes blood and it converts it into a number, right, for me. So that's media. And so as a media, this goes back to Jihoon's question also. I began to think of how this kind of media were technically and aesthetically composed. You know, when you think about the Burmese strain, the dangerous strain is in red. Very simple, but that's an aesthetic grammar, right? The people in the lab will not see it that way, right? So I was they got very curious about what I was bringing to the table, because I kept thinking them, this is a composition. There's an object, there's an environment. This is how we think about composition. So there was an exchange, and they began to see why certain media that compose my infection for me can have consequences, right? If I only think COVID was about me and my experience of COVID, and okay, I got the vaccine. I'm not an anti-vaxxer. I'm so grateful for the vaccines, right? But that's where the story ends, right? The diagnosis is the problem because I have lost the long view. So I think media studies and multi-species studies really helped get me to this kind of the way in which we think about media. It tells us many things. It tells us about capture, but it tells us about entanglement, right? And media studies has a very rich way of thinking about these things. So method-wise, in terms of choosing sites of research, Hejin, I started with the EcoHealth Alliance. What were they doing in Bangladesh? What were they doing in Congo? These were field scientists, and I followed it back to the labs where all this was being projected. That was my method in choosing those. So I don't know if that answers your question, but that's one. The other question that you had is how I got to this. And it's interesting because, when I wrote Global Icons, my second book, I was already thinking of the sensory. This is before affect theory. So I was looking at icons as, semiotic things. Somebody becomes an icon because you have cultural memory of that person. But you also have an affective and sensuous relation to the icon. So the sensory and the affective have always been with me for a very long time. I was interested in perception, how we perceive things. And then when I began to think about epidemic media for me, this became absolutely central. So I've been building and now extending a theory of apprehension, as not just fear and anxiety, but also, intuitive grasp of something that's not quite comprehensible yet.
Thank you.
All right, so other such comments or questions. I'm not sure whether all the people who registered came here, but I saw from the registering list, the people not only from cinema and media studies, but also from cultural anthropology, science and technology studies, East Asian studies, and art history. So I guess that there are other such questions coming from these different approaches or disciplines.
Cultural anthropologists have been really important to me in this book.
Yeah,
So
yeah, sure.
you're right,
Yeah.
the medical anthropologists, Andrew
Absolutely.
Lakoff, Melinda Cooper, all of this,
Mm,
all
mm,
of these people. Yeah.
So another thing that comes to my mind is your emphasis of the concept of apprehension that throughout your talk and your book, but one the tendency of the recent media theory is a kind of post-phenomenological turn of how media technologies affect our sensibility and of the world, in the sense that various computational systems or the autonomous devices, such as the sensors, RFIDs, or sensing devices, etc. Their operation and their transmission of information operate beyond the threshold of human perception. So it is necessary for us to rethink or conceptualize the ontology of media in ways that go beyond the traditional phenomenological understanding of perception and perception, prehension, or retention, built on the phenomenological perspective of human consciousness. So that the Mark Hansen's idea of the 21st century media, for example, demonstrates this point. In the base of the proliferation of those technologies in atmospheric ways that affect human perception, human perception, and human sensibility of the world, but also the ways that beyond the grasp of human perception and consciousness. So your emphasis on the apprehension in this book is interesting in this context. So I was wondering how can you respond to this conceptualization of the computational database or algorithmic media technologies and their uses in the medical or epidemic area in relation to that theoretical term?
Yeah, that's a really interesting question. I think with AI, this is going to become even more the case, right? That what we think about perception and its uses or its consequences won't be about humans anymore, right? So in that sense, I think that was where probably my humanistic training came in. I was really thinking about how our epidemics, and I would say COVID shaped a bit of it, because I finished the book during COVID, and it was it became really about what does it mean to live through an epidemic, to live with it, to survive it. So it was, in that sense, you're right, it is still human-based, the perception. Now, the technologies I deal with are much more than that, right? So, and that's why I'm interested in all this noise, the things that we don't look at, right? There's so much that is captured, but we don't even understand, actually, right? It's particularly with the inscription of viruses, right? You actually don't understand. So one of the labs that I dealt with was, they've been making one model of HIV for 40 years. Yeah. So, and they're still doing research on it, right? So this tells you that they have huge amounts of data, right? And one of the things they do is they try to crowdsource this data and produce these models, right? Now, what I was interested in, so I kept saying, there's nothing we should not know, but we actually can't process it. So there is, on the one hand, there's a sheer volume of information that's coming in. Some of it is not actually, it's just noisy. We don't know what it is, right? And I think with AI, this is just going to increase, right? There's going to be things that we are told that we will not understand, right? So you're absolutely right in that way, that why then focus on human perception, right? And modes of human perception. And I would say, because for me, the thing I was trying to get to is, how do we as humans perceive epidemics? Because we are the ones who try to come up with all kinds of solutions, right? And the solutions we have come up with, like pesticides or medicine, overuse of medicines, right? The pharmaceutical empire, have been completely damaging. So I was going after a certain kind of damage that came from human perceptions and their limitations, when we have so much technology that we can actually go to for more. Yeah. But I would say, Jihun, my book is not in concert with the kind of the techno-perceptual, analysis. Yeah, it is much more in that way, it's more social science oriented. It's in critical global health studies. Yeah.
Thank you. All right. So any, question or comment?
It's
May
a lot
I
for people
ask?
to take it. Yeah. Yes.
This is very superficial and sketchy question, but I was wondering whether the specific characteristic of media, for example, PCR, are expressed in the visible way or perceivable way. I mean that polymerase chain reaction is very specific in its exponential amplification, and there are many specific characteristics of this media. For example, you can use it in criminal forensic, and so on and so on. So are those kind of characteristics expressed in the way we can perceive, or since the content and form of this media is so segregated that we cannot perceive it? This question is in the context of structuralist films, because they try to represent the form of film into the content of the film. So yeah, that was my question.
Really interesting. I have to think about the structuralist film part. But I would say that a media technology like the PCR it does many things, right? So like even the camera trap technology, all of these have multiple uses. I think what I was after is what particularly are you looking for, and how does that shape what you compose, right? So if infection, right, your state of infection is the most important thing, then you will count not only the viral strains, but you will do a ratio, right? X amount of virus and Y amount of blood. That's a particular form. The ratio form is a particular kind of form, and that's important for infection, right? Because then, as you know, with HIV, it's undetectable or not, and so on and so forth. Sorry, did you want to say something else? Yeah. So I guess certainly PCR is used for, criminology, etc. But the question you would ask is different, right? And maybe the form in which your result, I'm thinking of it as, like a movie is a media form. I'm thinking of the PCR test result as a media form. Are you looking at a ratio, or are you looking at something else, right? So that is one of my answers. That certainly you could say many uses of the PCR. Your comment about structural film is actually interesting, because I've never thought of it in relationship to the PCR, and how the form of it is already there in the way in which the reaction is actually plotted. Yeah. I have to think about it more. But what would you say? I'd love to hear what you're thinking, how you're thinking about the relationship.
I think there is a certain process or circuit of kind of mingle those two, content and form.
Yes.
And for example, polymerase is the very factor that enables chain reaction.
Yes.
So
oh, I see. You're going there. Okay.
Yeah, I think in molecular biological sense, although I have to work more on it, there can be significant relationship between its expression and its process. So, but I don't think that all media is the same, symmetrical, but I think that's why it's used in so many laboratories, that it helps us protect and predict for more epidemics in universal dimension. So yeah, that's my idea.
Yes. That's it's an interesting idea. I'll still keep thinking about it. But I take your point that what you're doing is taking a bit of the DNA and amplifying it, right? And so in that way, the form, you've already identified a form, and that is going to give you the content, right? So in that way, I take your point that there's something structural going on. It's a structural form that we know from the film analysis. And I also take your point that I wonder how far the comparison will go. Yeah. But what I like about your question is that, there is something comparative in terms of process. And I think Professor Kim brought up the processes of mediation, right? There's something in the processes of mediation that are comparable. Like if you're doing a nature documentary, right? And you're following an animal with a camera, right? There's often things that are very unprecedented that happens, right? So there are some processes across media that compare. But your question is an interesting one. I hadn't really thought of comparing it to film. So thank you. Yeah.
So any other questions? Actually, so the questions, made by, Mr. Kwon, also, also made me think of another question, but so I reserve it for other, audiences who come up with your own. So the previous conversation about PCR also reminds me of how data visualizations are perceived from different perspectives. Probably that's also related to how a component of epidemic media works in the society on different scale or different perception of knowledge. So we have seen so many, data visualization related to death curve and the spread of pandemic and or the transnational, or translocal, distribution or spread or the pathogens, in the pandemic situation. So we have so many different types of the ways in which the visualizing the impact of the virus or on human beings and the environment on different scales. And but the data visualization themselves, I think, are subject to two different, perceptions or understandings. One is, definitely, they serve as a kind of the illustration of the knowledge about the virus as a kind of knowledge obtained by mechanical or computational, instruments. So it serves as a kind of the token of objectivity, but as Lorraine Deston and Peter Garrison have demonstrated in their, rich study of the change of the idea of objectivity in the history of science, the uses of mechanical tools for demonstrating objectivity also necessitate or involves the existence of trained observers, such as medical specialists or the specialists of different sciences, in order for the visualization to be acknowledged or authorized as mechanical, or the objective, evidence. But in watching, various data visualization in different media, people's reaction to them are beyond transcend that kind of truthfulness, even expanding into various objective responses, including all feeling sublime, the feeling awe and anger, etc. So these two different responses are part of how pandemic works and how the epistemic media works and how it related to human sensibility in relation to the existence of the causes of dangers whose scale go beyond the human perception and understanding. So yeah, my thinking is that the epidemic media involves not only different scales and different spatial temporal relations or different entanglement of human and non-human the elements, but also different reactions and different ways of affective and intellectual responses. So that's just a thought that I had in listening to the conversation prior to, related to prior question.
Yeah, I mean, that is interesting because I do address that a little bit in the book where, you know, there are things that are made by scientists for other scientists, right? So that's the mechanical objectivity, let's say the hubris. But actually, if you look at how scientists work, they build this model and they sort of the visualization of the virus is actually because they want to put all the data together and visualize something as a hunch. So they often talk about, you know, this is an intuition that if we put the molecular data together with the cellular data, this is what we'll get. This is what it looks like. But the actual thing is a kind of is a hunch or a proposition to other scientists. So I find them actually really aesthetic and creative in all kinds of ways that Peter, Laurent Aston and Galison have said, mechanical objectivity. And then there's a bunch of, you know, epidemic media, which I think after the COVID experience, we've learned a lot about how to read, you know, the rising curve, what it means. I didn't know what we didn't know what flattening a curve was. We learned that. Everybody knows that, right? For instance.
Yeah.
So I think that in the book, I talk about as mesoscale literacy, sort of the middle scale, you know, not the expert and not the general, you know, understanding or reaction, but a mesoscale literacy. And a lot of HIV/AIDS counselors and doctors really try to do that. You know, they teach you to understand your blood test as a ratio. What does that mean even? Right. And so that I think there's a lot of work around mesoscale literacy that's very interesting. Yeah.
Yeah, actually, the one thing I forgot to mention before the start of this event is that this fascinating and rich, you know, event was preceded by, you know, the coincidentally, you know, Anna Ching's, you know, talk on her, you know, the martyrdom at the end of the world on the occasion of its translation in Korean. Just it took place before 2 hours before the event. So some of the audience here might have the opportunity to attend the two events that are close to, in many ways, you know, close to each other. So I hope that today's event and the Bishnu's talk, you know, provide you with the opportunity to expand or to reconfigure the idea of media in broader and deepened ways. And the next Cinema and Media Studies Colloquium event will host, you know, the UC Parika with his new book, Operational Images, in early December. So we'll publicize the event as the time is approaching with my social media and other, you know, channels. So please stay tuned. And so so let us thank again for Bishnu for, you know, sharing her research as well as her insight for comments and thought, you know. So please, you know, give her a hand.
Thank you for hosting this, June. Appreciate it.
Yeah, all right. So let's keep in touch soon. And so
yes.
Yeah.
Yes,
so
okay.
Have a great
Bye.
evening. Yeah,
Bye. I'll see you. Yeah. Thank you. Bye.
Bye. 예, 마치겠습니다. So